Histidine is my favorite amino acid. It's like the beauty queen of amino acids in that it's always changing its looks, and it can never decide, like, it doesn't want to be neutral, it doesn't want to be positively charged. Instead, it goes back and forth, and this allows it to do cool things like serve important enzymatic roles, helping speed up reactions. In its neutral form, it can also bind to metals, which makes it really useful for purifying proteins through using like histag technology. So I want to talk more about histidine today, but first let's look at, okay, well, how do we recognize it?
Histidine is really versatile, and this versatility can actually be really useful, but it can also make histidine kind of hard to recognize because there are different ways that we can draw it. So one of these depends on whether or not it is positively charged, and this is going to depend on the pH. I'll get much more into this in a minute, but histidine has a pKa of around 6, which means that at a pH of 6, you're going to have about equal numbers of it be protonated and positively charged and deprotonated and neutral. And so above a pH of 6, less than half, and the further you go above the pH of 6, the fewer there are going to be that are protonated. In our bodies, we typically have a pH of about 7.4 or so. So we're looking at somewhere between 1 to 10% of the histidine in our bodies being in this protonated state, and the rest of it being in this deprotonated state. This imidazole. So we call this imidazole, and we call this imidazolium ion. And so an ion is a charged thing. This is positively charged, so we call it a cation. This positive charge is actually going to be kind of shared between the two different nitrogens, and we can see the charge drawn on either one of these nitrogens. And so we call these resonance structures, where basically we can draw these in two different ways. So here we're varying where we put the charge, but both of these nitrogens had a proton. Well, now what happens if you go to a higher pH? At a higher pH, you're not going to have enough protons for each of the nitrogens to have one. So a high pH corresponds to a low proton concentration. So if there's a low concentration of these protons, it's going to be harder for the histidine to be protonated. So less of it's going to be deprotonated, more of it's going to be deprotonated. And when we talk about deprotonation, we're only losing one of the protons. So one of these nitrogens is still going to have a proton, but which one? It could be either, and therefore we can draw that proton on either of these nitrogens, and we call these different tautomers. And so you can think of both of these tautomers can exist under certain conditions. One might be more favorable than the other, but you'll see them both. And so lots of different ways we can draw this histidine. And all of this was involving what's going on at these nitrogens in terms of their protonation. But none of that is affecting the actual aromaticity of the histidine ring itself. So histidine is aromatic. When we talk about aromatic amino acids, we typically focus on tryptophan, phenylalanine, and tyrosine. And the histidine, although it's aromatic, that aromaticity gets outweighed by its ability to be basic and to basically have a positive charge. So we call it the amino acids that have a positive charge. We call these basic often, because what happens is a base is something that, and one definition, a base is something that accepts a proton. And so histidine in its neutral form can accept a proton and act as a base, that is, and become positively charged. On the other hand, we have acidic amino acids, which an acid is something that donates a proton. So in their neutral form, aspartic acid and glutamic acid donate a proton to become their what we call conjugate bases, aspartate and glutamate. And in the case of the basic amino acids, here we have things that are acting as their neutral form acts as a base. And so if you act as a base, you take a proton. And so what histidine can take a proton and become the positively charged version. And it's more likely to be, these are more likely to be positively charged at a lower pH where you have a higher proton concentration. And so in the case of histidine, where it's going to have a pKa of around 6, and the pKa corresponds to the concentration of protons, at which half of it is going to be protonated and half of it is going to be deprotonated. But at a lower pKa, you're going to have a lower pH, you're going to have more protons, so it's going to be more likely to be protonated. And at a higher pH, you're going to have fewer protons and you're going to have less of it be protonated. Because it's like a log scale and stuff, what happens is that for each increase or decrease of one pH unit, you get a tenfold change. So when we're looking at these amino acids like lysine and arginine, they're basically always going to be in our bodies, they're almost always going to be positively charged because we're way above the pKa. With histidine, we're actually a little below the pKa. So we're going to have significant amounts of both the charged and the non-charged, but the charge but the non-charge is actually going to dominate because we are going to be at a pKa of around like 7, so we're above the pKa of histidine, which means that there are more protons available, fewer protons available, so more of it is going to be in the non-charged state, which is going to be important, as we'll see, because then it can act as a nucleophile. So histidine's charge is going to dominate what we talk about often when we're talking about histidine, but it still is aromatic. And so this aromaticity, what am I talking about with this? Well, I talk about it a lot more in my posts on like phenylalanine in particular, as well as these other ones. But basically this means that in addition to sharing electrons between neighboring atoms, those electrons are kind of shared between all of these carbons in this ring. And so we refer to when you have, so a single bond, basically when you see those individual lines, this represents a single bond, which is a sharing of one pair of electrons. If you see a double line like this is a double bond. This is 2 pairs of electrons that are shared. When you have aromaticity, when you have like resonance, these electrons are shared between multiple atoms. And when you have aromaticity, this sharing happens in a ring. And so basically what happens is that they have a little extra electrons, so they have enough for a single bond, but not quite enough for a double bond. And so they share that extra into a kind of communal pool that these all can use. And so it's not really that you have these alternating single and double bonds, but instead it's something that you have this electron, it's something in between where you have a partial double bond character between all of these. This is going to have in order for this to happen, you need to have it like the electrons have a certain configuration. You have this electrons kind of above and below the plane. And in order to line all their electron sharing clouds together, you basically have to have them planar. And so these electrons, they have to basically, when we talk about like sharing pairs of electrons, it's really more like they're kind of like syncing, like overlapping regions of their electron cloud. So where the electrons hang out in these like clouds. And these have like different shapes of orbitals, places in those clouds where the electrons like to hang out. Much more complicated than I can go into here, but in the case of this resonance, we're talking about these p orbitals, which kind of form these hourglass structures above and below the plane. And so you have to have these hourglasses line up, and so you can't like bend your ring or anything, because then you would be making those hourglasses not line up. So basically, bottom line, aromatic molecules are going to be planar, they're going to be flat, and they're going to have electron density spread out above and below the plane. And electron density is the electrons, those are negatively charged. So you're going to have a concentration of negative charge above and below the plane. And this is going to help allow histidine to bind to metals, which metals are often cations, so positively charged. And we'll get more into that in a minute. It's harder to see with histidine that you actually have aromaticity going on. With histidine, what happens is that in addition to like alternating single and double bonds, what happens is that you can also have resonance when you have like a lone pair of electrons participating. And so in these cases, even with whether or not you have the charge, you're going to have a lone pair of electrons is going to be contributing to this resonance. So you can kind of think of a pair of double bond, a lone pair of electrons as kind of being one of those types of double bond things. I know that sounds weird, but basically this, you can, if you push these around, you can like push the electrons here to make a double bond here, to make a single bond here, to push the electrons here. If you're able to kind of push the electrons with pushing electrons, if you've done OCHEM, this might make sense. If you haven't done OCHEM, don't worry about it. But bottom line is that these electrons are participating in this resonance. And another one of the pairs of electrons is going to be, could be used to form a bond to a proton. But see, this one still has a double bond attached to it. So this is kind of just showing you that it's still involved with that resonance. So all of this resonance is taking place, and you're still able to protonate or deprotonate without affecting that resonance. So histidine, no matter its charged state, is going to be aromatic. And so this is going to be important, and it can also be kind of confusing. So if you were to say, look at this, here you can see when we draw it like this, both of these lone pairs, if they're bound to a proton, are going to be involved with resonance. If you have only one of these is bound to a proton, well, this one that's bound to a proton, one of these lone pairs is going to be involved with resonance, so it's going to be non-reactive. But this other one is going to, that doesn't have a proton. Now those protons that could be used to form a bond to a proton are now going to be available to do other things, including form a bond to a proton, or it can act as a nucleophile. When something acts as a nucleophile, it can attack an electrophile, something that wants electrons, and we can get new bonds to form and things like this, and we can get you can get some weird stuff like histidine phosphorylation. So quick note that acting as a base is really just a special case of being a nucleophile. And so a base is just what we call a nucleophile when what it's stealing is a proton. And typically when we use the term nucleophile, if something is kind of like attacking something other than a proton and forming a new bond, rather than just kind of stealing a proton. But both of these cases are something acting as a nucleophile. The base is just a special case. And in both of these cases, with histidine, you're going to need that lone pair of electrons to act as a nucleophile, whether it's in a traditional nucleophile sense or whether it is acting as a base and stealing a proton. And so for both of these, you're going to need it to be deprotonated. And so this is why it's really important that you have a significant amount of histidine in the deprotonated state in your cells, so you have that lone pair available that can do that nucleophilic attacking. And also that you can go back and forth between being protonated and deprotonated relatively easily at our body's pH. But I'm not going to go into that very much here. Only wanted to say that when you have histidine, you're going to have it be aromatic. This is regardless of whether or not it is protonated. When you have protonation, or you're basically always going to have one of these nitrogens be protonated. At a higher pH, you're going to, at a lower pH, you're going to have both of these be protonated. At a higher pH, you're going to only have one of them be protonated. And in all of these cases, the histidine is going to be aromatic, which means that you're going to get that delocalized electron density above and below the ring. So histidine's ability to go back and forth between having two protons and having one proton, this is going to be really, really important. And especially its ability to have significant amounts of both of these present at a physiological or bodily pH. So although our pH in our body is going to be above this pKa, it's not that far above. And so this means that we're going to have, say, even if you had like 5% of it be in this protonated form, that's still going to be significant enough to have important functions. And even if you have most of it in this deprotonated form, well, now it can serve important functions in this form as well. And what's important is that it's able to go back and forth fairly easily in our bodies. And this can be useful in, say, the active site of enzymes. And so enzymes are reaction speeder uppers. They're often typically protein, sometimes protein RNA complexes, sometimes RNA alone. And they're able to kind of help make reactions happen. And they can do so in different ways by holding things together and the right orientation. And one way that enzymes can speed up reactions is by giving and taking protons when you have like an acid or base dependent reaction. And histidine is able to give and take protons, as we saw. And so it's going to be very helpful for a number of different reactions. One example is with RNase A. So RNase is going to be an RNA cutter. It's a nuclease. And how RNase A works is it actually has these two histidines in its active site. So in the place where things are happening. And in the case of this RNase, what's going to be happening is that the RNA is going to be getting cut. You have a kind of like ping pong mechanism where you have one histidine that's going to be deprotonated and one histidine that's going to be protonated. The deprotonated one is going to steal a proton, and this is going to then cause the RNA to attack itself, and then that attacked RNA is going to then attack the other histidine and steal its proton, and then that was going to attack water, and that's going to back attack this. And so basically you get this kind of like ping pong mechanism where you have this hydroxyl, this OH group, be activated on the RNA, making it nucleophilic and much more nucleophiles than other posts. But this is going to then seek an electrophile, and it finds one in this phosphate group, and it's going to attack. And when it attacks, then you're going to get this broken, but then you get this awkward intermediate. And so then this histidine is going to activate a water molecule, and then that water molecule is going to attack this and free it in this normal form. And all of this is taking place because you're able to do this ping pong mechanism, giving and taking protons from histidine. And so often in the active site of enzymes, you're going to have an altered environment that's going to make it more conducive for either giving or taking protons under various conditions and various things like that. So histidine is really, really cool. And RNase A, not so cool in the lab. So we often have to try to inhibit it, such as by using molecules like DepC. And so much more on this in my post on RNase A. But histidine is often going to be crucial in enzymes. Another way that histidine can help out in enzymes, as well as in the lab, is by allowing it to bind to metals. So we already talked about one way that histidine is able to attract metals, and this is through its aromaticity, because it has the delocalized electrons above and below. And this is going to allow it to attract the positively charged metals. So often metals are in what we call a cationic or positively charged state. And metals are going to, you would think, so when we think of histidine, we think of it being positively charged, it's like, okay, well, that doesn't seem like that would be very likely to bind a metal. But when histidine is binding metals, it's going to be in its deprotonated state. So you don't have that positive charge. And you have this electron delocalized above and below the rings, which is going to be a kind of like nice landing pad for a metal. And so what happens is that you can get these attractions to metals, and then histidine is able to do another thing. It's able to form what we call a coordinate covalent bond. So in a normal bond, you have neighboring electrons. One atom is going to share one electron, and the one next to it is going to share another electron, and they're going to form a bond that way. But you can also get a bond form, what we call a coordinate covalent bond or a dative bond, where one of the atoms is going to donate a pair of electrons, and the other one's just going to like come along for the ride. So one of them is going to donate all of the electrons in the bond. And this can happen if you have an atom that has a lone pair of electrons that can share. And if we go back and we look at the histidine, when histidine was in that deprotonated state, well, now it's going to have a lone pair of electrons that it can share, and it can share those electrons with a metal in order to form one of these dative or coordinate covalent bonds. So this can be really, really helpful when you're binding a metal, because these metals are also going to be useful for enzymes because they have these big electron clouds that can kind of give and take electrons, and they can help coordinate things bound together and really, really useful. And so histidine is going to help you be able to hold on to metals, which are going to be able to let you do other things. Often though, just having one single bond to a metal isn't going to be enough. Instead, what you want is something that's going to act more like a chelator. So a chelator is something that's going to bind down on a metal in multiple places. So it's going to take multiple chelates like claw, and you can kind of like think about how dentate your chelator is. But so we talk about claws, chelate, and then dentate, so like teeth. How many bites is it taking? Where each of these bites is going to be one of those coordinate covalent bonds. If you have a single histidine, it can only take one bite. But if you have a lot of histidines in a row, well, now you can take multiple bites. So you're polydentate. And then when you have something that can take multiple bites, we call it a chelator. So sometimes we see these chelators in molecules, like in the molecule hemoglobin. It's going to have these 4 binding sites. It's going to be tetrabentate, and bind down to iron. Or you can, in the lab, we often use EDTA, this chelator, this molecule that's going to be able to form multiple binds to magnesium. And in the lab, what we can also do is we can make histidine into a better chelator by attaching a lot of histidines in a row. And if we add this onto the end of a protein, then we can make it so that this protein has a chelator attached to it. And it's going, so that will allow it to bind to metals. And if we then flow this protein through a column containing these little beads or resin that are coated in a metal, say nickel, well, now our protein can get stuck to the column. We can wash everything else off, and then we can use a competitor to basically push off the protein. So we get our protein to bound. We have our protein with what we call, we'll talk about this in a second, but this like his tags. We have a string of histidines in a row. So like 6 or 8 typically. You have these beads that are coated in nickel or another metal such as cobalt. You flow the protein through. Your protein, because it has that chain of histidine that's going to be able to act as a chelator, it's going to stick to the beads. Everything else is going to flow through. Now that you've got all that other crud washed off, you want to get your protein off. And so now you can basically add free imidazole. So this is just like the free part of the histidine. So not the whole amino acid, but just this ring part. You can use that as a competitor. You have to add more of it because you don't have a bunch of them in a row, but this is going to allow you to then compete off your protein. And the reason why this works is because although proteins naturally have histidine, they don't naturally have like 6 to 8 of them in a row. So you're not going to have this chelation effect as strongly. But in order to get your protein to have it, you have to do some molecular cloning. So basically, when you're introducing the instructions for making the protein, say into bacteria cells, you can alter the recipe so that it has the instructions for making histidine. So histidine is coded for by CAU and CAC. So basically, if you put that sequence in the front of the instructions for making a gene or for making a protein, what's going to happen is that then when the ribosomes of the protein making machinery goes to make that protein, they're going to start making it or finish making it, depending on if you put it at the beginning or the end of the protein recipe. They're going to be adding extra histidines in. And so they're going to be adding those chains of histidines. So your protein is now going to be tagged. It's now going to have this, what we call like an affinity tag, that you can then use in order to purify it, because the other proteins, although they might have some histidine, they're not going to have that many histidines in a row. So we can do this in either like a gravity flow format, so just like with resin, and that you pack into like a plastic column, and then you drip things through with gravity, or you can use a machine like an ACTA, an FPLC, a fast performance liquid chromatography, or fast protein liquid chromatography, depending on where you look, that basically pumps it through the columns. If you're using one of those ACTAs, you can then set up like a gradient elution. So I talk a lot more about his tag purification in another post I'll link to, but basically you can either just kind of wash everything off and then go stepwise and go to a high concentration of imidazole, or you can do a gradient where you kind of gradually raise the concentration of the imidazole. So remember, the imidazole is our competitor. And so because you have some natural proteins binding, so if a protein just like happens to have a lot of histidine, it might bind to the column. And so you want to kind of make sure to prevent non-specific binding. So basically that sort of thing. You typically include a low concentration of imidazole, somewhere maybe like 50 millimolars or something like that, 25, 50 in your background, just to like prevent any non-specific binding. And then later you're going to add a higher concentration to push your protein off. If you can do this stepwise, where you basically just go right away to a higher concentration, maybe like 150 millimolar, or you can do a gradient where you gradually increase the concentration, and this can allow you to kind of separate things that might have, if you have some things that are more weakly binding versus strongly binding. Various things you can do with that, and I'll link to the post on my Histag purification post where I have a lot more detail. In addition to binding nickel, histidine can bind other metals, including cobalt. So sometimes you might see like cobalt, NTA, you might see like talon resin, things like this. Basically, cobalt is going to be pickier, and so it's going to have a higher specificity for binding to your histag protein, but it's going to have a lower affinity. So you're going to need more of it to bind the same amount of protein. And again, more on it in my post on histag purification. But the histidine is going to allow us to do this because histidine is able to form those coordinate covalent bonds. And those coordinate covalent bonds are going to let it bite down on metals, which we can use to purify, or we can use it in our bodies, basically to just hold metals in place and help out reactions. One thing that's not great about histidine, though, at least if you have allergies, is that it's the source of histamine, which is basically this molecule that's involved with like an allergic response. So histamine, amine, this is NH3, or an NH containing group is an amine. And what happens is the histidine, so we have a carboxyl group here, it's going to get removed as carbon dioxide. And now you just have that amine group. And so this is histamine. And this conversion is done by histidine decarboxylase. Another way to break histidine down is by using histidase, which removes the ammonia group. There are a variety of ways you can break down histidine, and you end up with glutamate. And this glutamate can then enter the tricarboxylic acid cycle, which means that it can be used to make glucose. So we call histidine glucogenic. As you might have guessed, based on having that positive charge, at least sometimes, histidine is going to be hydrophilic, and so you're often going to find it on the surface of proteins. So that's the basics of histidine. It can be protonated or deprotonated, and unlike the other basic amino acids that we see, the ones that can sometimes be positively charged, histidine is actually more predominantly going to be in its neutral form. But it can easily go between this positive and this neutral form, giving and taking protons. And this is going to allow it to serve important functions in helping make reactions happen that involve giving and taking protons. And so histidine, you often see it being something like a ping pong mechanism, where one histidine gives and one takes, and then they give back and take back, and some really cool stuff like that. It's also going to be aromatic. Regardless of whether or not it's protonated, it's going to be aromatic. This means that it's going to have electrons delocalized above and below a plane. So histidine's ring is going to be flat, and there's going to be kind of like a donut of electrons above and below it, which is going to help it bind to metals. It's also going to be able to bind to metals through coordinate covalent bonds, thanks to having a lone pair of electrons in this deprotonated state that can then form a coordinate covalent bond to that metal. And this is going to allow it to act as a chelator, so something that kind of like bites down on metals. If you have multiple histidines, you can get these like chelation effect where you have multiple bites on a metal. This is going to allow it to us to use a string of histidines as a histag in order to purify proteins. So lots of cool things about histidine, and now you know more about it.